Well, very welcome to IFOM. So today, the message I would like to convey is that the study of the fundamental mechanisms impacting on DNA repair and genome integrity might be beneficial for precision medicine, okay? And also it's an occasion to tell you what we're doing. Let's start with this. So these are chromosomes from cancer cells, which are completely scrambled, as you can see here. And this is typical of all tumor cells. And that's the reason, the genome instability, why cancer cells evolve, and it's the reason why they avoid apoptosis and why they become resistant to drugs. Now, all these kind of rearrangements, like translocation, inversions, and deletions, and so on, have a common molecular cause, which is called DNA recombination. Now, DNA recombination is very important during meiosis. It's important to fix DNA lesions, but it's very dangerous during the mitotic cell cycle, okay? And the reason is that unscheduled recombination during the mitotic cell cycle generates all these kind of problems. So normal cells suppress recombination during mitotic cell cycle. Tumor cells have too much recombination. Now, this is what's happened at the somatic level in every tumor, but you can entirely recapitulate all these problems in cells coming from these patients. These are very rare syndromes. These are called genome instability syndromes, which are caused by mutation in different genes. All these genes encode enzymes involved in DNA repair. All these patients have something in common. They have cancer predisposition, but also they have premature aging. This is a Japanese lady, 12 years old, 48 years old, okay? And also neurodegeneration. Why all of this? It's unclear. It's actually much more easy to understand why they have cancer predisposition, because the DNA is mutating all the time. But why this is causing premature aging and neurodegeneration is still unclear. In any case, the take-home message of all these studies is that the biggest problems to elicit these unscheduled recombination events is during chromosome replication. And DNA synthesis during S phase is very dangerous, because it's a potential substrate for recombination. And most of my talk will be focused on this kind of topic here, and I'll tell you a little bit more about these regulators of DNA damage. So what we're doing, well, our lab is working on chromosome replications in many years, but more recently we have been studying the key regulators of the DNA damage response, and namely ATR and ATM. These are PI3 kinases, very similar to mTOR, which are the apical regulators of the DNA damage response. They control check1, check2, but also p53, and also apoptosis. So in our field, the idea is that ATR is sensing DNA lesions in terms of single-strand DNA, while ATM is sensing DNA breaks. Although I would like to challenge this kind of concept, and particularly I would like to challenge the concept that these proteins are only tumor suppressors. Well, I say it's obvious to think that might be tumor suppressors, because if the cells are mutated in this protein, in these genes, they should be unable to coordinate cell cycle with DNA repair options, and therefore they should accumulate mutations. And that's why we think about them as tumor suppressors. But this is part of the story. There are genetic syndromes caused by mutation in ATR or in ATM, SECL in AT patients. This is a very rare syndrome because ATR is an essential gene, and these patients carry hypomorphic mutation, while ATM seems to be less essential. In any case, again, you have progeroid syndromes, neurodegeneration, and cancer predisposition. If you knock down conditionally the ATR in mice, it's obvious the phenotype. It's a progeroid syndrome, really, okay? So premature aging at every level. And the reason is because they lose stem cells. So what we did in many years was to study the contribution of ATR and ATM in controlling the integrity of replicating forks. What you see here is a replication fork. Just focus on the most important thing. These forks have always 3 branches, 1, 2, and 3. Even if you challenge DNA synthesis with DNA synthesis inhibitors, some of which are chemotherapeutic agents, or with DNA breaks, the cell has always to maintain 3 branches, okay? And the reason is that ATR and ATM, and ATR also in this pathway here, all they do, through the actions of many, many enzymes, is to prevent the conversion of 3 branches in 4 branches, okay? Why this is dangerous? Because 4 branches at the replication fork resemble a holiday junction. I don't know what you know about molecular biology, but holiday junction is a cruciform structure, which is the most important intermediate in homologous recombination. So if you don't have these enzymes, you end up generating substrate for recombination in S phase. And from now on, you degenerate completely because you generate inversion, translocation, and so on, chromosome instability. So that's why we believe these are key factors involved in preventing recombination and tumor suppressors. Now, this is so important for cancer therapy because, as you know, most of the anti-cancer drugs, the classical one, but also the new drugs like PARP inhibitors or ATR inhibitors, at the end affect DNA synthesis. DNA synthesis is the most sensitive moment for the cells when you challenge cells, cancer cells, with chemotherapeutic agents. So we have been doing a lot on this kind of field, including profiling these drugs with the genetic backgrounds and so on. But we also decided to approach the problem by another point of view, more basic, and that is ATR at the end is an essential gene. And normally, in our body, we are not exposed to chemotherapeutic agents or to DNA damaging agents. So we decided to address the more fundamental role of ATR in the cell. And turns out that still is during S phase. But during S phase, lots of things happen which can be dangerous. So you have to think about the chromosome like a truck in which different trains collide, the DNA polymerase train and the RNA polymerase train, because you have lots of transcription in S phase. Now, the collision between RNA polymerase and DNA polymerase is a huge problem in S phase, and is the most genotoxic event that might happen. The coordination of this event is crucial, and ATR is coordinating this. All the problem is at this level. This might be complex, but just to tell you that it's like a rope. Every time you open and you make the DNA polymerase going through the DNA helix, you generate downstream lots of torsional stress, which is called supercoil, that must be resolved thanks to topoisomerases. Needless to say that topoisomerase inhibitors are very popular in cancer therapy. But that's what they do, okay? But the most important picture here is that this torsional stress at the end generates energy, accumulates energies, and generates mechanical stress. And this is the mechanical stress that, if it's not properly controlled, can lead to aberrant transition of the chromosomes, including the formation of those cruciform structures. And what we showed here is that ATR is playing a clear role here in coordinating and preventing these events. Why does it do that? How? So we have been working for many years to try to understand which is the sensing mechanism. But the answer came from a colleague of ours, Nancy Kleckner, who nicely showed that ATR is a fantastic protein able to sense mechanical stress. It's like a spring. And this is what it looks like, okay? It is like a spring in terms of, enzyme. But what Amit did in our lab was to show that ATR has another property. This is the nuclear envelope, and this is the chromatin which is attached to the lamine. Every time there's a torsional stress at the level of chromatin, this stress can be transmitted to the nuclear envelope, to the lamine-associated domain. Now, the beauty of the nuclear envelope, it is that it is an elastic connector, so ideal to sense mechanical stress. And ATR, even following subtle changes in chromatin dynamics, relocalize at the nuclear envelope to deal with this kind of molecular torsional stress. And this is published. Now, keep in mind that the envelope is connected also to the plasma membrane, to the cytoskeleton. And it's also true that if you pinch the plasma membrane, you transmit mechanical stress to the nuclear envelope. And ATR can sense this. In this case, you see here, we pinch with patch clamp the plasma membrane, and in green you see the ATR from the nucleus relocalize it to the nuclear envelope. And this happens within seconds, which is consistent with a mechano response. So obviously, this kind of discoveries led us to a different field. So I told you we started with DNA, we started with the replication of fork integrity. I'm telling you now that ATR is a mechanosensor, okay, which is able to sense mechanical stress generated by cytoskeleton, okay, but also by the chromatin dynamics. This is a picture showing interstitial migration. These are cells which migrate into pores. Typically, this is what cancer cells are doing during metastasis, like to squeeze into pores. Now, I don't know if you know that, but the nucleus is the stiffest organ in the cell. So during migration into pores, the nucleus has to squeeze, the chromatin has to condense. And this is a regulated process. So the elasticity of the nucleus is crucial, okay? So ATR defective cells have loose elasticity, okay? I will stop here with two take-home messages. So the plasticity of the nucleus has a huge impact in terms of controlling chromosome integrity, but also cell migration. And ATR, and I can tell you that ATM plays a similar role, although different, together with another complex, which I haven't told you, TOR2, which is mTOR with a different flavor. They are controlling the integrity of the chromosome, but also they couple genome integrity with cell plasticity and cell migration. This work would have been impossible without the contribution of mechanical engineers that work in the lab and biophysicists that are able to measure these kind of events, okay? And this is what we're doing a lot in IFOM, and this is thanks to the people in the group and the contribution of these people, and also to the mechanomedicine program, which we established a few years ago in collaboration with the Mechanobiology Institute of Singapore. IFOM has a main branch here, but also has a location in Singapore with whom we work on this kind of topic. I will stop here. Thank you for your attention.